Hello everybody and welcome to this exciting new webinar entitled Accelerating Oncology Drug Discovery with Label-Free Imaging. My name's Dr. Emma Gibbin and I'm the Communications Specialist here at Nanolive. And I'm very excited to share some of these new results with you today. So on this first slide you can see an outline of today's talk. I'm going to begin by briefly introducing today's topic and show you the advantages that Nanolive imaging has over traditional techniques. The majority of the webinar will be spent showing you the results of a case study that we conducted in-house, where we exposed both pre-adipocyte and cancer cells to various kinase inhibitors that have shown promise as anti-cancer drugs. These screens yielded several novel observations, and so I encourage you to stick around to the end of the webinar to enjoy these. I will finish as always by answering 3 of your questions, so if you do have a question, please enter it in the box at the bottom of your screen. If I don't have time to answer your question today, then I will personally respond as soon as possible, so don't hesitate to contact me. So without further ado, let's get started. So we all know that cancer is a huge problem in our society. Over 100 different cancers are known to affect humans, and it's the second leading cause of death in the world. The World Health Organization estimated that 9.6 million deaths were caused by cancer in 2018 alone. Treatment traditionally has been focused on chemotherapy to treat cancer patients, but the non-specificity of this approach often has detrimental side effects. As such, there's great interest in identifying or creating small molecules that selectively target cancerous cells, and this spawned the drug discovery. So there's 4 steps in the drug discovery process. The first is to identify a valid biological target, characterize their structure, and then create a range of candidate drugs that may be capable of inhibiting the target. The second step is to test these drugs on cell disease models in culture. This step must be completed before the drugs can be tested on animal models and then finally enter clinical trials for humans. So why is Nanolive imaging advantageous compared to traditional imaging methods? Well, this is the traditional approach to in vitro screening. Cells are manipulated and cultured in 96 well plates. The candidate drug, be it either an inhibitor or an antibody, is then applied. The cells are left to respond for a certain amount of time and then they are fixed. Fluorescent dyes are added if required, and finally the cells are imaged. All of this work produces one single discrete endpoint, data point. If additional time points are required, then the whole experiment must be repeated using different cells. So endpoint data such as this has several limitations. Firstly, it's extremely time-consuming. It requires a huge amount of manpower. The reagents can be expensive. But perhaps most importantly, the methods themselves can change the response of the cell to the drug, so you never know if what you're measuring is a response of the cell, is a real biological response, or simply a response to a method. With Nanolive imaging, the process is a whole lot more streamlined. So cells can be cultured, perturbed, and imaged throughout the experiment. Avoiding the use of fluorescence means that we measure the natural response of the cell to the drug rather than an experimental artifact due to phototoxicity or the use of a fluorescent label that can interfere with the observed phenomenon. It also means that we can measure cells at high resolution for long periods of time without damaging them. The result is continuous data that captures dynamic changes in morphology with high temporal resolution. So let's show you this in action. As I said before, we will focus on kinases as anti-cancer targets. Why? Well, protein phosphorylation controls almost all of our cellular functions. Metabolism, cell proliferation, apoptosis, and inflammation are just a few examples. Phosphorylation is controlled by two enzymes, kinases and phosphatases. Kinases catalyze the transfer of phosphate groups from high-energy phosphate-donating molecules to specific substrates, while phosphatases act in the opposite direction. The human genome contains over 500 different types of kinases. Hyperactivity, malfunction, or overexpression in kinases is the root cause of many human cancers, as we can see in this table on the right. The development of specific kinase inhibitors has therefore generated great interest as an anti-cancer treatment. One of the benefits of targeting kinase is that selective inhibitors can be created to target specific pathways. We identified 8 inhibitors or modulators of kinase activity. Our targets were AKT, JNK, CDK2, RTK, PKA, JAK, mTOR, and AMPK. We conducted 2 screens of the kinase inhibitors. In the first screen, we used 3 T3-derived pre-adipocyte cells, and in the second, we used glioblastoma cells. Experimental conditions were kept identical between the 2 screens. So cells were always plated on fibronectin-covered dishes, and they were kept in serum-free conditions. The inhibitors were used at 3 concentrations, at 0.1, 1, and 5 times the recommended concentration that we found in the scientific literature. We included pre-adipocyte cells in the experiment, not as an example of normal cells. In fact, these cells have been immortalized and are thus very similar to cancer cells in their capacity to proliferate. Rather, we included them as an example of a less aggressive cell type, with the hope of observing different phenotypic responses to drugs. So both screens were conducted using Nanolive's automated microscope, the CXA. Images were acquired at an acquisition rate of 1 scan every 25 minutes for a total of 40 hours. Unfortunately, I don't have time to show you all of the results from these screens, So instead, I've chosen to highlight some of the most interesting findings over the next couple of slides. I will begin by showing you the negative control. So if we start the video here, we can see that from the segmented image on the right, that the cells started to relatively low density. If we zoom in on one of the cells, we can see that the cells are in good shape. The nucleus, nucleoli, nuclear membrane are clearly visible and have a normal morphology. Actin cytoskeleton is spread evenly across the surface of the dish. There are lipid droplets, the bright spots that you can see in the cells present, and the mitochondria are elongated. As the experiment progresses, we see cells dividing and the cell population increase. Because the cells are in serum-free conditions, they are essentially starved, and so they resort to metabolizing their lipid droplet reserves, which are finished around the 30-hour mark. After this point, the cells begin to start dying. So one of the advantages of our techniques is that it captures all of these dynamic details that would be missed without live, label-free imaging. And these are important details to note when we compare the response of cells to drug perturbation. The first treatment that I want to show you is mTOR, is what happens when we inhibit mTOR. So mTOR is a key regulator of cell growth and proliferation. So this video on the right, you will see that it coincides with a graph showing cell area. And one of the first things you notice when I start to play is that the cells are much larger than in the control. The second thing that we can notice from this treatment is the formation of autophagic vacuoles in the cell cytoplasm, shown here. This process begins approximately 5 hours after the inhibitor is applied and is swiftly followed by cell death. Just see that one more time. Okay. The second treatment that we will focus on shows the response of cells to JNK inhibition. So the JNK pathway is a death signaling pathway that can promote apoptosis in either the nucleus or the mitochondria. The first thing I want to point out before I start the video is the debris that you see in the background of the image. I want to reassure you that this is not a sample preparation issue. Rather, these are focal adhesion points where cells have tried and failed to attach to the surface of the dish. So you'll see when I play the video that indeed the cells in this treatment were highly motile. The graph on the right shows cell count. So when we start the video here, one thing you can notice is that this number stays relatively stable throughout the experiment. So when JNK, the death signaling pathway, is inhibited, cells do not divide, but neither do they die. The second unique feature is being shown on your screen at the moment, and that is the distribution and localization of mitochondria. Normally mitochondria tend to cluster around the nucleus, but here they are distributed throughout the cell and are even present in the lamellipodia. The final very interesting thing to note is that even towards the very end of the experiment, as we're approaching now, lipid droplets are still present right at the very end, suggesting that JNK inhibition interferes with cellular metabolism. The third treatment I will feature is the CDK2 inhibitor. CDK2 stands for cyclin-dependent kinase 2, and it drives the progression of cells into the S and M phases of the cell cycle. And so we expected CK2 to have severe disruptions within the cell. So if I start to play the video here, the first thing that you will notice on your screen is that cells have notably thicker nuclear membranes than in the control or in other treatments, and structurally disorganized nucleoli. Cells do divide during the experiment. There's an example in the top part of your screen here. but they don't follow, they don't look like normal process. So normally with our technology, we can see clearly defined stages of mitosis, chromosomes lining up on the metaphasic plate, etc. But here the process resembles more apoptosis before division occurs. The last thing to note here is that again lipid droplets are present at the end of the experiment, showing that CDK2 inhibition again causes metabolic alterations in the cell. So this table gives you an idea of the type of novel information that can be assembled from CXA screens. So here I went through every individual video, analysed the changes in cell morphology that occurred in response to each drug. I did this for both pre-adipocyte cells and cancer cells, and then created this table to compare the phenotypic responses of the different cell types. So we can do this in both a quantitative and a qualitative manner. Where there's a quantitative analysis being done, you will see the arrows next to the symbols, and the direction of the arrow shows the effect of the inhibitor relative to the negative control. For example, where there's a green arrow, it shows that cells will have a greater response or a more enhanced response than in the negative control. So the first thing I want to point out is that glioblastoma cells are clearly much more resistant to kinase inhibition than pre-adipocyte cells, which we expected. Glioblastoma is one of the most aggressive forms of cancer. This is why we chose to focus today on the response of pre-adipocyte cells to show you clear treatment effects. The second is that phenotypic responses can also be cell-specific. pKa, for example, reduced cell size in cancer cells, but had no effect on pre-adipocyte cells. So there's so much novel information here, and this is from 2 simple screens, that this type of approach is guaranteed to generate new hypotheses. So I do want to still show you the response of cancer cells to a certain treatment. And one thing that we can do here is to show you the effect that different dilutions of an inhibitor had on the glioblastoma cells. So to illustrate this point, I will focus on the treatment that initiated the greatest phenotypic response in cancer cells, which is CDK2. As before, we'll begin by showing the negative control. So if I start to play this video, the first thing you might be able to notice is that the cells are more motile than the pre-adipocyte cells in the negative control. They divide a lot more frequently. The cell population more than doubles throughout the experiment. And the last thing to notice that differs from the pre-adipocyte negative control is that lipid droplets were still present throughout the experiment. And we see no cell death even towards the end of the experiment. So I'm going to start to play the response of the video from the CDK2 inhibition. And the first time I show you these videos, you may well think that the cells resemble that in the negative control. It's true that the morphological response of CDK2 is much less pronounced than the drastic nuclear changes we observed in the pre-adipocyte cells. However, cell metric analysis revealed there are subtle concentration-dependent shifts in cell size. The histogram in the bottom shows the distribution of cells according to their cell area. At 0.1 times the lowest concentration, cells are positively skewed. At 1 times the distribution becomes more normal, and at 5 times we see the emergence of a long tail on the right. And then we can run all of the videos at the same time to show you this shift in cell size occurs in response to concentration, in response to the concentration of the inhibitor that's provided. So information such as this can be very informative for identifying appropriate dosage levels of drugs. So in conclusion, Nanolive imaging validates known functions of cells in a quantitative manner. Our technology is label-free and non-invasive, and this ensures that our results are unbiased and reports the real response of cells to a drug perturbation, rather than the response of cells to method perturbation. This increases the biological relevance of your results and should inspire confidence in your data, in the data that you produce. It also permits a huge range of novel observations to be made, including, but not limited to, the formation of vacuoles within the cell cytoplasm, the temporal dynamics of lipid droplet use, changes in the distribution or localization of mitochondria, and changes in the nucleoli and nuclear membrane, which are simply observations that are not possible to make with any other technology. So all of this is possible with Nanolive CXA. So what are you waiting for today? Get your quote and book your demo today at the website on screen and accelerate your drug discovery program. So I hope you enjoyed the presentation, and now I'm going to answer some of your questions. So the first question is, what type of 96 well plate are compatible with the CXA? So Nanolive had already developed specific well plates to fit the microscope that have the correct optical properties and that are fitted with a lid to ensure that there's no evaporation and that environmental conditions such as temperature, pH and humidity are as stable as possible during the experiment. Second question. You mentioned the negative effects of fluorescence. Have you tested for this? So yes, one of my colleagues, Hugo, did his master's thesis on exactly this topic. He compared the possible phototoxic effects that were induced by Nanolive imaging with those that were induced by epifluorescence imaging, and found substantial differences between the two. He found that mitochondria were particularly sensitive to phototoxicity. So if you're interested in reading Hugo's master's thesis, it is available to download on our website. So I encourage you to do that. It's a really well done study. Finally, the last question is, what is the maximum number of experimental conditions that can be run simultaneously? So for this, it's possible to automatically image up to 60 well plates, 60 different experimental conditions in well plates in parallel. Of course, there is a trade-off between the number of conditions that you run and the temporal resolution of your experiment. So I hope that helps. I hope you've enjoyed the webinar. If it will be available later to download, so if you want to see the results in more detail or to watch the videos again, then I encourage you to subscribe to that. But for now, I'll leave you there. So thank you very much for joining me, and I hope to see you soon. Bye-bye.